Hi, I'm Ruoshi Zhang from the Max Planck Institute of Biophysical Chemistry. Here I will give a brief introduction to SpacePHARER, our tool for sensitive identification of phages from the CRISPR spacers in the prokaryotic hosts. More and more bacterial phages, which are viruses of bacteria and archaea, are being sequenced from the environment. Annotation of these phages also requires identifying their hosts. Conveniently, the CRISPR-Cas system, an adaptive immune system that exists in bacteria and archaea, carries the information of previous phage infections. CRISPR spacers are short DNA fragments of phage origin. By matching these spacers to the phage genome, we can reveal phage-host relationships. Previous methods that directly blast the nucleotide spacer sequence against the genome of phage can yield reliable predictions, but at a low sensitivity. Also, the low search speed is hindering annotation of many phages and hosts usually found in the metagenomic data. With this in mind, we developed SpacePHARER, which can identify phage-host relationship at higher sensitivity and speed. In SpacePHARER, we first 6-frame translate all the spacers and phage open reading frames. We then search the translated spacers against phage proteins using MM62. Next, we calculate a combined score for every host phage pair, with all significant spacer hits given their p-values, which represents the strength of evidence. Lastly, we make de novo predictions, that is, we do not rely on previous knowledge by performing a downstream FDR analysis and report the most confident phage-host pairs. We think performing the search on protein level can improve the sensitivity because phage tend to mutate their DNA sequences while keeping the protein sequences conserved. Also, multiple spacers can target the same phage, hence combining the hits will improve the prediction. We compare the performance of SpacePHARER to the state-of-the-art method using BLAST-N. The false discovery rate was calculated for each of the methods using a different no-model dataset in the FDR control step. We show that at common FDR threshold of 5%, SpacePHARER predicted 3 to 4 times more phage-host pairs than standard BLAST-N, while being around 50 times faster. The speedup allows us to analyze large-scale genomic and metagenomic data. SpacePHARER can also make host predictions at a better precision than BLAST-N by a weighted LCA procedure using the MM6 taxonomy module. If you're interested, please check out the poster about the MM6 taxonomy module also from my group. SpacePHARER is available as a free open source software. It is also possible to install from Conda. For more details on how to install and use the software, please visit our GitHub repository. Please feel free to drop any questions below in the text box. I will be happy to answer them. With that, thank you for your attention.